When most people look at a pond, a lake, or a stream and think about how it functions, maybe how green it is, how much photosynthesis is happening, or even how many fish are going to be produced by it, they're probably not thinking about viruses. But over the last several decades, there's been an increasing amount of attention paid to the role that viruses play in aquatic environments. So, to address that role, I'm going to briefly introduce what a virus is, talk about some hypotheses that describe where viruses might have come from in an evolutionary sense, talk briefly about how viruses are believed to affect aquatic ecosystems, and then to cover some viruses that are important to understanding aquatic ecosystem ecology. So, what is a virus? A virus is an infective particle that is made up of, contains hereditary materials, right? The chemical building blocks of biological information, which can occur as single-stranded, double-stranded DNA, or as RNA. And that's important for identifying groups of viruses. Likewise, there's a protein coat that surrounds this hereditary material. It protects the hereditary material and can also be used in identifying viruses and groups of viruses. Sometimes there's a lipid coat that surrounds that protein coat as well. So that's what makes up the infective particle that we refer to as a virus. Viruses, as a part of aquatic ecosystems, were recognized when people started to think about things that were showing up on very small, pore-sized filters. And so as people were studying aquatic environments, they were noticing that there were these small particles and that viruses were making up some of the small particles that they were collecting in aquatic ecosystems. It's important as we think about viruses to remember that there are viruses that affect every branch of cellular life. So there are viruses that affect bacteria, archaea, and eukaryotes. Eukaryotes being the large domain, the group of organisms that's most like us and includes humans. As we talk about viruses, I might also mention virions. These are viruses that are outside of living cells. These are viruses in the environment. And in aquatic ecosystems, and in particular, as we've started to understand the role of viruses in aquatic ecosystems, we know that there's a large reservoir of virions outside of host cells. It's also important to think about viruses and their small size. If you think about the physics of this situation, they have a small Reynolds number. And a Reynolds number, in short, measures the forces of inertia relative to the forces of viscosity. And as a result, it's fairly easy for these tiny particles to not sink because they don't have a lot of inertia relative to the viscosity of water that they're in. Viruses are extremely common. We find them virtually everywhere. And we know that they can outnumber bacteria by an order of magnitude or more. And it's been hypothesized that they have more influence on how many bacteria die, when those bacteria die, than do the protist predators that we're familiar with. The small eukaryotes that are swimming around in aquatic ecosystems. And so it may be that viruses are a really important part of aquatic ecosystems, and we should work on understanding them. So I mentioned that viruses infect all branches of the tree of life. And that's not to say that any one virus affects all those branches of the tree of life. Rather, that there are viruses that exist that take advantage of each of those branches of the tree of life. So this is a very common way of representing the relationships between those branches on the tree of life, and viruses are everywhere. So, if viruses affect all the branches of the tree of life, where would they have originated? For a lot of people, that suggests that they have a very ancient origin. And there are three hypotheses that have been presented in the literature that talk a little bit about where viruses might have come from. So, one of those hypotheses is the virus first hypothesis. The idea that hereditary materials might have organized in a way that they could take advantage of cell-based life. And that viruses might have existed first. Another hypothesis, the escape hypothesis, is the idea that maybe this is a bit of cellular machinery that went awry, that broke off, and now does its own thing. And it can replicate itself, not using its own tools, not using its own cell, but rather it can replicate itself using other cells. So we can kind of think of that as rogue machinery. Or, the third hypothesis is that viruses might represent a reduction of a line of cells. Basically, this idea is that if a line of cells specialized in parasitizing and using other cells, there might have come a point in their history where those viruses no longer needed the rest of their cells because they were using a host cell. And so they stopped producing their own cells. So those are three hypotheses that describe how viruses might have come to be. And remember that in science, that's always changing, right? If there's better data, if there's a better explanation for the data that we have, then we would have to change that explanation. I mentioned that over the last several decades that the idea of viruses affecting aquatic ecosystems has become more and more important. So in the 1970s is when people started to recognize viruses as very common in aquatic ecosystems, and the number of publications that describe viruses in marine and freshwater systems has increased since then. And this paper is a few years old, but it captures that trend. So, how is it that viruses affect aquatic ecosystems? Well, to really understand how many active viruses there are, we need to be able to determine whether those are active or whether those viruses, because of the environment, have been deactivated. More about that in a few minutes. And so one way that we could do that is that we could infect targets. We could infect host cells with a diluted series of these viruses and see how many of those actually do a good job of getting into the host cells and using them. And so we could extrapolate out the number of active viruses based on the dilution factor and the number of infections produced. As we start to think about how those viruses are going to impact the living systems there, we need to think about that group of viruses or virions that's present in that aquatic ecosystem and what happens to those virions. So here's an image from a paper that I used to prepare this video, and it shows viral particles as well as cells. So one of the important things that we need to understand how viruses are going to affect that system is how stable those viruses are in the system and how stable the system itself is. So viruses could get pushed around by the movement of water. Viruses could be broken down and deactivated. It's also important to think about how frequently a virus is going to interact with an appropriate host cell. So if it's unlikely to encounter a host cell before something breaks its chemical machinery, then it's pretty likely that that group of viruses in that location is going to be reduced over time and is going to go away. On the other hand, if hosts are common, then there's a pretty good chance that those viruses are going to be able to encounter those hosts, infect them successfully, and increase in number. One of the important pieces of that encounter rate is how many different hosts a virus can use. Right? So we can think about that as host breadth. We might also care about how much the virus impacts the host. Basically, does it kill hosts? Does it use a host and allow that host to go on living? So does an infection mean that that host is removed from the pool of appropriate hosts in that location? And as we think about that, viruses that affect single-celled organisms are probably going to have a much greater impact on those organisms than those viruses that affect multicellular organisms. Why is that? Well, if that virus is lysogenic, that means that if that virus is the kind that causes a cell to break apart to release the new virions, that it's going to kill that whole organism if it's a single-celled organism. Multicellular organisms might have mechanisms that can reduce the damage that it experiences. And so that organism might be able to survive even if the virus is the kind that breaks cells apart. So viruses can be lost from that pool of virions in the environment through a couple of mechanisms. UV light can break them down, right? Can disrupt their structure such that they can't infect new cells. The presence of improper hosts might affect that encounter rate enough that those viruses don't get a chance to infect the appropriate host before they're deactivated. And viruses might be eaten up by some of the other organisms that are living in that environment if they're small enough to handle the virus and take it in. So a couple of important viruses in the aquatic environment include the bacteriophages. This is a broad group, a broad sort of categorization of viruses, and it's defined as viruses that infect prokaryotes, viruses that affect the bacteria and the archaea, which are some other groups that you can find information about in some other videos. These might be the most abundant viruses in aquatic ecosystems. And they come in various sizes. There are miniphages and megaphages. And it's interesting that they have different population dynamics in shallow waters where they're considered evanescent. They sort of blink, populations of these viruses blink in and blink out of existence, which probably tracks their hosts, but also the physical environment. But in deeper waters, they're persistent. We find those viral groups over and over again in the same environments. And like I said, that virion abundance probably tracks host abundance, and in a lot of cases, that's actually been demonstrated. It's also important to think about that ability for viruses to replicate using a host cell, and then what happens to that host cell afterwards. There are viruses that are considered chronic. That is, they use the host cell for a long time and continue producing viral particles and don't immediately kill the cell. There are also lysogenic ones where those viruses infect, they replicate, and then they cause that host cell to break open and release the virions back into the environment. So viruses have been recognized as an important biological compartment and have been recognized as deeply influencing the way that aquatic ecosystems work. And so I'm providing you a quote here, and I'm going to read a couple of the ways that this paper captured other papers as having described viruses in aquatic ecosystems. In some cases, they've been described as partners in pelagic foodwebs. So the pelagic is the open water part of those foodwebs. They've played important roles in killing the winner. And what that means is that there's a density-dependent effect for some groups. So if you have a more common organism, it's more easy for those viruses to infect and damage it. They've been described as new players in the game and major players in global ecosystems. So there are hypotheses that place these infective particles as a really important part of the functioning of aquatic ecosystems. To the point that there's a term, the virioplankton, that's been added to some models of aquatic food webs. And what that means is essentially that there are these floating potential virus, potential infective things in the water column, and there's this pool of materials that's suspended in the water. There's dissolved organic matter in the water, some of the stuff that can change water colors towards brown, etc. But in that stuff there are viruses, or potential viruses, and those viruses are constantly infecting the phytoplankton, the floating photosynthesizing organisms. They're constantly infecting the bacterioplankton, all those suspended bacteria in the water column, regardless of what they do, how they make their living, or what their role is in that ecosystem. And those viruses are also impacting grazers. And so, as a result, that virioplankton, that suspended group of virions, is going to affect the dynamics of phytoplankton, bacterioplankton, and grazers. And there's this idea that because of the way that viruses work and the way that they influence their host, that there's the potential for more dissolved organic matter, DOM, and more particulate organic matter, POM, to be released into the environment because of the activity of those viruses. So I think the point here is that, one, it might favor heterotrophic organisms, those organisms that eat other things that don't produce their own ways of storing chemical energy, right? The autotrophs, those are things that photosynthesize, that make molecules that can store some energy for later use, right? Like trees, like algae. But the viral shunt might favor heterotrophs, those organisms that have to eat other forms of carbon that can then be used for energy later. And that's likely to affect the way that aquatic ecosystems work. Another way that viruses influence aquatic ecosystems, and in a way that's probably more familiar to a lot of us, are diseases like viral hemorrhagic septicemia. So this disease has been identified as a disease of fishes, and it was described quite a long time ago in Europe and has been tracked there. More recently, it was identified in Canada around the year 2000 and in the US around 2005. And so here's one example map that that shows infected waters, which includes in Wisconsin the Lake Winnebago system, Lake Michigan, Lake Superior, and probably includes the major river systems as well. We know that viral viral hemorrhagic septicemia is dangerous to at least 25 species of fish. Many of those are sport fish, including muskies, yellow perch, and brown trout. And there's been no described danger to humans. So remember that viruses are often pretty specific, and we've got a lot of distance between humans and fishes. We're very different creatures in a lot of ways. And so it makes sense that those viruses are unlikely to affect the humans directly. When we look at fishes that are affected by VHS, we see symptoms like hemorrhaging, bulging eyes. Those frequently scale up into unusual behaviors and eventually kill the fish. So we know that the virus is shed into the water through urine production, reproductive fluids, and can also be taken up into new hosts by fish that eat other fish, so piscivory. The virus can remain active in the water for a significant amount of time, so it can be active and potentially infect new hosts as a virion for at least 14 days, possibly longer. One of the biggest recommendations by agencies who are dealing with this is don't move dead fish, don't move live fish, don't collect minnows in one system and then use them in another system, because that's a great way to spread viruses. And you might not know if those fishes are infested yet or not. So today, if you got all the information that I was trying to share in this video, you should be able to describe the physical components of a virus. You should be able to contrast that with what you know about living organisms. Viruses are considered non-living, so if you think about what's in a cell, you should be able to compare and contrast the two. You should be able to list and explain those three hypotheses describing where viruses come from. You should be able to talk a little bit about the likely effects of viruses on ecosystems, and then you should be able to describe bacteriophage as well as viral hemorrhagic septicemia as important parts of aquatic ecosystems. Thanks for listening. I hope you have a great day.